Look at the sheer volume of data generated by a modern sequencer. Lasers scanning flow cells output billions of reads in a single run, but that massive computational power faces a severe physical bottleneck. The exact moment that a biological sample is separated from a human body, the tissue begins to degrade. Collection, shipping, and isolation introduce pre-analytical variables. These are the critical vulnerabilities where enzymatic decay and environmental noise ruin the raw material before downstream bioinformatics ever sees it. Achieving high-confidence diagnostic results requires absolute physical and chemical control of the sample before it ever reaches the laboratory. The ONEOMICS product ecosystem is architected to manage this exact vulnerability, serving as a vertically integrated genomic supply chain. This catalog outlines the entire pipeline. Looking at the bottom right, we see the end goal: complex diagnostics like MyOmics, MyNIPT, and MyGutBiome. To reach these endpoints, the process begins at the top, with precise collection of the initial product. To extract flawless digital data out of a sequencing pipeline, you must perfectly preserve the physical biology going in. Biological data acquisition begins at the point of care, requiring immediate chemical intervention the second a sample is drawn. The sample collection and preservation section illustrates halting biological degradation at time zero. The OneSpit system provides a mandatory stabilization buffer. Historically, the primary logistical and geographic bottleneck in global multiomics has been the cold chain, the absolute reliance on dry ice and ultra-low temperature freezers. If dry ice sublimates during transit, or a mechanical freezer faults on a tarmac, the biological sample is lost before extraction can occur. The alternative to this risk is the deployment of ambient room temperature stabilization chemistries. The storage and shipping section isolates transit degradation. Ambient guards lock unstable nucleic acids, cells, and proteins into suspended animation without freezers. This global advantage allows mRNA therapeutics to ship without ultra-cold infrastructure, utilizing compact temperature regulation only for specific edge cases. Breaking the dependency on dry ice logistics unlocks economic scalability and expands reach to clinics that lack specialized infrastructure. Ambient shipping logistics decentralized advanced molecular medicine, making complex diagnostics accessible regardless of geographical location. The pipeline now transitions into the laboratory. Perfect upstream preservation yields nothing if the downstream extraction process is inefficient. The core biochemical challenge is separating pure target nucleic acids from the complex tissue matrix and from the aggressive stabilization buffers introduced during transit. The rapid DNA and RNA isolation section details the suite required to execute that exact biochemical separation. Notice the shared 1-mag prefix across the different sample types, indicating the core methodology utilized here. This relies on magnetic bead-based extraction. The beads bind specifically to the target nucleic acids, allowing contaminants and cellular debris to be completely washed away. However, a single universal extraction protocol will fail when encountering complex environmental samples. That is why the catalog lists tailored kits specifically engineered for non-clinical matrices, like soil and plant. Soil and plant matrices contain highly specific PCR inhibitors, like humic acid or complex polysaccharides, that will outright blind a sequencer if they are not aggressively filtered out during isolation. Tailored matrix-specific isolation acts as the pipeline's final purity checkpoint. It ensures only high-fidelity molecular material enters the sequencer. The culmination of this entire biological data supply chain is the final clinical diagnostic readout. Looking at the services section, we see diverse, high-level diagnostics relying on the preceding pipeline. The accuracy of gut and vaginal biome sequencing is directly linked back to the fecal collection kits used at the very beginning. The viability of non-invasive prenatal testing depends entirely on cfDNA preservative tubes preventing contamination in transit. Clinical exome and rare disease testing require ambient guards to prevent molecular degradation during the journey. The success of these algorithms relies on the physical material fed into them. These diagnostics are the emergent properties of a flawless physical supply chain. High-confidence clinical outcomes demand an unbroken ecosystem from the patient to the final data output. In modern biotechnology, diagnostic truth begins the exact second the sample is collected.